Hi, and welcome to the Johns Hopkins Cerebellum seminars. Today, as our guest, we have Shlomi Haar. Shlomi earned his undergraduate and master's degree in biomedical engineering from Ben-Gurion University of the Negev, and then a PhD in brain and cognitive sciences, also from Ben-Gurion. During his PhD, he was mentored by Ofer Donchen, where he studied encoding of arm movements in the human brain. In 2017, he joined Imperial College London as a Royal Society International Fellow with the Brain and Behavioral Lab, where he was mentored by Aldo Faisal. His fellowship research was on the relationship between changes in the brain and the body of learning of novel, real-life motor skills in an attempt to uncover what makes some of us better learners. In November 2020, he moved to the Department of Brain Sciences to start his own lab as an Edmund and Lily Safra Research Fellow at Imperial College. His research program focuses on improving our understanding of neurobehavioral mechanisms of motor control and motor learning in health and disease, particularly in Parkinson's disease. Shlomi, good to have you with us.
Thank you so much, Reza. So the cerebellum in Parkinson's is the topic for today. And well, Reza, thank you for the introduction. Just to add, well, first of all, a disclosure. I'm one of Reza's many scientific grandkids. As Reza mentioned, I started my work with Ofer Donchin, where I joined his lab during my undergrad and did my master's recording cerebellar interneurons, and that's my first paper. I continue for a PhD with Ofer and Ilan Dinstein, doing MRI recording, looking at simple single joint movement like most of our field is doing. And during my PhD, I was getting the urge to move away from the simplistic movement to full body, real world behavior, and went enjoying Aldo Feisel's lab at Imperial with the Royal Society Fellowship to look exactly on that. That was interfered by COVID, which got me engaged in clinical data science at Imperial at the time and pushed me towards more translational clinical application and led me to take the Safra Fellowship at the Department of Brain Sciences, trying to take a more translational route of the insights from motor control, motor learning towards Parkinson and neurodegeneration. And that's what I've been doing for the past 5 years. I'm happy to inform you that this summer I'll be moving to the University of Surrey, where I was appointed as a senior lecturer, associate professor, and my group will be moving there later this summer. So I'm working mostly on Parkinson's for the past few years. So briefly on Parkinson's, as I'm sure you're all aware, Parkinson is a neurodegenerative movement disorder. It's considered a movement disorder due to the motor symptoms. Tremor is probably the most famous one, but actually not the most dominant one. Bradykinesia, the slowness of movement, and rigidity, the muscle stiffness, are actually the more common and more severe motor symptoms. But Parkinson is not only motor, there are many other motor and non-motor symptoms. And in fact, if we are looking at the trajectory of Parkinson, time zero, the time of the diagnosis, is the beginning of the motor symptom. And it's considered a movement disorder because that's the things that we recognize. The motor symptoms are what we see, and that's what lead to the diagnosis. But actually, Parkinson starts many years before, and the non-motor symptoms are developing over that time with the neurodegeneration before the motor symptoms first appear and the diagnosis comes with them. And there is a question of what's going on during this premotor, this prodromal period, and why other symptoms start to evolve while the motor symptoms are waiting longer. And could the cerebellum be involved in it? So with that in mind, let's take a step back and talk about the anatomy of Parkinson. So Parkinson is a neurodegenerative motor movement disorder. The neurodegeneration is happening in the substantia nigra, in the basal ganglia, and it's mostly the dopaminergic neurons, and therefore for over 70 years, Parkinson is being treated with levodopa medication, basically giving the body dopamine. And the Parkinson patients receive dopamine, and when it kicks in, their symptoms are under control, and when the medication washes off, their symptoms return, and Parkinson a clinical specialist, and all the treatment are about optimizing it to control the fluctuation. So that's different treatment with the medication, with pumps. Deep brain stimulation is also, all of it is helping to modulate those fluctuations because it's all about pushing the dopamine in the right order. Thinking about the computational neuroanatomy of the Parkinson disease, we have a classical textbook model, which is very well explained and very well known. Briefly, the direct and indirect pathways of the basal ganglia are both being affected by the lack of the dopamine. This leads to under inhibition of the globus pallidus interna, over inhibition of the thalamus, and therefore lower excitation to the cortex. So that's the classical textbook view of a circuitry of the basal ganglia. And what's the cerebellum? Does it link to it? How might it be linked to it? So typically in this classical model, we don't link the two. And if we go back to the computational neuroanatomy of motor control, I assume you're all familiar with that figure from Reza and John from 2008. So that's papers that I was raised on. And we're talking here about two parallel loops, a forward model loop through the cerebellum and a cost and reward through the basal ganglia. So two independent loops. Actually, I was so influenced by these papers that Ofer and I produced a bit more than a decade later a revised version of it where we mostly talked about more parallel loops. We talked about the different cortical regions having their own parallel loop, but still neglected any connection between basal ganglia and the cerebellum. And we were clear about it, right? We were making it clear that the models failed to address findings that show direct connection between the basal ganglia and the cerebellum, and specifically mention here Boston and Strick. So the work from Peter Strick lab shows multiple connections from the cerebellum and from the basal ganglia with the cortex, and actually that was part of our motivation, our intuition about the existence of multiple parallel loops and different cortical areas being supported by their own parallel loops with the cerebellum and the basal ganglia. The things that we neglected from the model is the fact that they also show direct connection between the basal ganglia and the cerebellum. Well, thinking about the model of the basal ganglia, a few years later, a group led by John Rothwell were suggesting that this connection between the cerebellum and the basal ganglia are linked into the basal ganglia circuitry, our classical direct, indirect, hyperdirect, but that makes the model much more complicated, much less intuitive to follow and to understand how it works in Alzheimer's disease. But we need to understand because it is important to the circuitry. So the cerebellum in Parkinson, as mentioned, Parkinson is considered to be a disorder of the basal ganglia. The neurodegeneration is in the basal ganglia, and most Parkinson research throughout the years focused on the basal ganglia. So this paper from 2013 about the cerebellum in Parkinson was basically stating that the cerebellum is overlooked. Yet they have found few evidence that suggests that there is a role for the cerebellum, both in pathological, there is a pathology of the Parkinson happening in the cerebellum as well, but also they suggested a compensatory effect of the cerebellum in Parkinson. So the pathological changes may account for several clinical symptoms, but the compensatory effect may help me maintain relatively normal motor function. And they have this figure, which I'm still yet to fully understand because we have parallel lines instead of connecting lines, but basically they suggested that there is some pathological effect, so a decay in the cerebellum that start in this preclinical stage and continue into the clinical. There is a compensatory effect starting at the preclinical stage, peaking at the time of the diagnosis and decaying over time. And there is also effect of dopaminergic treatments that increase cerebral function, and then it decays over time as well. So if we try to map this on the time course of the Parkinson, the important thing to note is that at the time of Parkinson diagnosis, what they claim is that there is a compensatory effect that's happening starting before that is peaking at this time of the diagnosis at the beginning of the motor symptoms. More recent view, this is 2013, linking the cerebellum to Parkinson, an update. So this update have a new graphic for the model of the direct, indirect basal ganglia loops with the connection to the cerebellum. Still, the image and the interpretation of this model becomes so much more complicated than our intuitive direct, indirect, hyperdirect. But once again, important to dig into and to try to understand. And specifically, the connections are going between the direct with the substantia nigra, but the most interesting connection comes from the cerebral cortex to the STN. Now, importantly, here they have in the review covering many studies that have looked at the cerebellum in Parkinson, showing cerebellar atrophy in multiple lobules, showing increase and decrease in functional connectivity, and also showing hypermetabolism in different lobules of the cerebellum. So much work have done that actually shows involvement of the cerebellum in Parkinson. A big limitation of all of it, if we want to address this initial hypothesis about the role in terms of compensation, is that the vast majority of the work was done on a cross-sectional cohort. So we're looking at Parkinson's early versus late, or a healthy versus Parkinson's, and very little work was done longitudinally to see how the progress is happening. And there is big data today, big cohort that enable us to look into such things. So the biggest cohort in Parkinson research is an initiative of the led by Michael J. Fox Foundation, the PPMI, Parkinson Progression Marker Initiative, that they have cohort of thousands of Parkinson patients, and they doing data collection every 6 months. They also have prodromal and healthy controls within this data set. So that's a huge resource that allow us to look into patient over time and to really do a longitudinal study. The problem is that while they do MRI scans, this is very sparse, and looking in this data, there are less than 100 patients with multiple MRI scans that allow us to look at them longitudinally. But less than 100 is still a very meaningful cohort. So, Jenna's master project was looking at the anatomical data from the PPMI longitudinally. And the interesting and important addition that she've done here was not only looking longitudinally at the Parkinson patient, but also looking longitudinally at healthy control. And that was taken from the OASIS data set. So if we're thinking about the progression of Parkinson, we're looking now at all the participants, patients and their match control at the participants, sorry, at the patients at the time point of the baseline, right when they were just diagnosed, their first PPMI visit, and again 2-3 years later. And the controls similarly were also taken at 2 time points with 2 to 3 years gap. And that allows us to look at the changes that are happening during those 2-3 years in healthy controls in that age groups of 60 plus, relative to the Parkinson patients. Therefore, accounting for any atrophy, any changes in the brain that are happening due to healthy aging within the OASIS, and compare it to the changes that we see in the Parkinson's patients. So, looking at the total intracranial volume, at our baseline, we've seen no difference between the groups. Then all the regions that we're looking at, and we're not looking specifically on the cerebellum. While I'm putting it here in the context of the cerebellum because, well, that's the topic of the seminar, but also that's the most exciting finding. We were looking in a data-driven approach on subcortical regions that involved with the motor symptoms, with the motor system, sorry, and we're not looking only at the cerebellum. But looking at the results, we see that we have significant difference during baseline between the Parkinson patients in brown, let's call it, and the control in blue, in the white matter of the cerebellum, right and left, and as well as in the thalamus and in the pallidus. In all cases, we had higher volume in the Parkinson cohort, so higher relative volume, suggesting that in all those regions, during baseline, right, at the time point of the diagnosis, at the beginning of the motor symptoms, we actually have higher volume in the Parkinson patient, suggesting potentially some compensatory mechanism that is leading to them being having higher volume than the healthy participant at that point of time. So yeah, and to highlight the cerebellum here. Now the next step was to look longitudinally. What happened 2-3 years later? So now everything here is relative. We're looking at the difference, at the change, at the decay that we have within the control and the patients over time. So when we're looking at the total intracardiac volume, we had decay in both groups, healthy aging, and actually bigger decay in the healthy control. The decay in the healthy control was significant, while the decay in the Parkinson's was there but was not significant. Now when we're looking at the relative decay in specific brain region, we see few regions in the cerebellum, thalamus, basal ganglia, and the corpus callosum that show significant group difference, that show significant decay. When we're looking for group difference, we see group difference in the white matter of the cerebellum, so both right and left cerebellar white matter. The decay within the Parkinson's group was significantly more severe than the decay within the healthy cohort. While we don't see any such change in the basal ganglia, we do see some similar change in the corpus callosum as well, but in the basal ganglia we don't see a more severe decay, a significant more severe decay in any of the basal ganglia region during that time window, while we do see it specifically in the cerebellar white matter. Now we went further and look at the different cerebellar lobules to try to identify where is the most severe decay, and if we're looking at the different lobules, right and left, white matter, gray matter, we see once again it's in the white matter, as we've seen when we're looking at the cerebellum as a whole, and specifically we see it in class 1 and in lobule 9. So to summarize that paper, what we found here when we're looking at the anatomy longitudinally, we see an increase in cerebellar white matter in Parkinson patients during the point that they are being diagnosed, potentially suggesting some compensatory mechanism, more connections of the cerebellum during that point of time, and then accelerated atrophy over the next 2-3 years, suggesting the pathology affects the cerebellum during that time window. Now, the next step, the second paper that I want to present, looked at the functional connectivity. So once again, we're looking longitudinally. And here, so that's work that started by Sabati during his master's, then taken over by Jenna, and finally Love. We were looking at the effect of medication. So we were trying to look at this aspect of the dopaminergic effect, and can we really understand what it's doing to the functional connectivity over time. So what we often have with Parkinson patients is that shortly after the diagnosis it's starting to get medication, and what this initial model suggests that this medication is increasing the cerebellar function again, and in that sense the cerebellum might be involved in the positive effect of the dopaminergic medication in terms of addressing the symptoms and delaying the progression. So in order to look at it, we look now at 2 time points. We look at the patients post-diagnosis before they start to receive medication. So the novo scan, the last scan that they had just before after they were diagnosed, but before they start to receive medication. And then we looked at them again a year, 2-3 years after, when they're already medication. We're looking at the long-term effect of the medication, of the fact that they were medicated over a significant period, and how it affected their functional connectivity. So now we're looking at the Parkinson cohort, the novo versus L-DOPA, And the important thing here is the treatment duration, which varies in our cohort between 14 months to about 40, with an average of 24 months, so an average of 2 years of treatment duration. So now we're looking at the functional connectivity between the different nodes of the cerebellum, the basal ganglia, and cortical regions of the motor symptoms. And looking at functional connectivity, for those of you not doing MRI analysis, when we're looking at functional connectivity, we're looking at the correlation between the time course of the MRI during within different regions, different seeds that we choose, over the recording of 5 or 8 minutes of resting state typically. And they are highly correlated because we have shared activity and mostly shared blood flow, which is what we measured in MRI. So typically when we want to do functional connectivity in a meaningful way, we should remove the global signal, and that's what we did here. And therefore, after removing the global signal, the correlations are much lower. But the correlations that stay there are really relationship between the changes within regions after accounting for any global changes across the entire brain that take effect. So we see we have clear correlations within each one of the region, very high correlations within the cortex. All of this is known and repeated in many, many studies. The interesting thing here is that we looked at the same patients 2 years later when they're medicated, and we're looking for differences between those two maps. Looking at those two maps, I think that you can agree with me that we don't see any major difference. The patterns look relatively similar, and importantly, everything is paired, so we can run a pair test between them, so we can take one minus the other and look at the differences. And you can see that we have some nodes that will present differences. Some of those differences even pass initial statistical tests. Those are the ones that you can see on the lower diagonal. That's what passed the test. But when you correct for multiple comparison, and we have many multiple comparison here, nothing passed multiple comparisons. And actually, if we look at the distribution of the p-values, we see that it's more or less uniform, and therefore those that pass significance are basically just a type 1 error. So when we're looking at the individual connection, basically the connectivity haven't changed much during those 2 years. So our next step was, let's go beyond the individual connection and look at the network connectivity. So now we can take all those regions within the cerebellum and all the connection between them and try to classify the changes across all those connections from the de novo to the L-DOPA. And similarly we can do between the different networks. And then we can look at the classification. So where we're asking, can we classify between those maps, the individual participant maps, when they are medicated versus when they were de novo? And can we do it within each one of those quarters, basically? So can we classify the connections within the cerebellum, within the basal ganglia, motor cortex, and cerebellum to basal ganglia, cerebellum to motor cortex, basal ganglia to motor cortex? The only significant classification, you can see the ROC here, is between the cerebellum and the basal ganglia. So the only network effect that we can find for changes in functional connectivity was not within the basal ganglia, not the basal ganglia to the cortex, but actually the cerebellum to the basal ganglia was the network change that we've seen within this cohort when we're looking longitudinally for the effect of the medication. Now, to convince ourselves with that, we thought to take another approach, and that was Lav's master project, looking at eigenvector centrality. Eigenvector centrality is a different approach for looking into functional connectivity. It's, instead of choosing region of interest, choosing seeds and looking at the correlation between them, we are looking at all the brain, all the vox cells, and the connection with each other, and we are finding nodes that have more connections, that are connected to more other nodes, and that have higher connectivity. And we're looking for those meaningful nodes. So we can look at the map of the entire brain. We remove the white matter obviously here. We can look at the map of the entire brain and look at the eigenvector centrality as the metric of how connected this node is. And then we can, once again, look at the difference between the maps, and the difference map is what we see here. the changes in those connectivity, in those eigenvector centrality mapping between L-DOPA and de novo conditions. So what we see here in hot colors is the areas where we have an increase in the connectivity when in the L-DOPA versus the de novo, and what we see in cold colors is where we have a decrease in the connectivity. And interestingly, we see 5 functional hubs that shows an increase in connectivity during the L-DOPA medication. Those marked as A, C, E, F, and K here. And if we look at C and E, you can see that both of them are in the cerebellum, and they're not only in the cerebellum, so specifically in cross-2. If we look back at Peter Strick's 2018, the injection to the cerebellum here is actually, well, we can look back to the 2010 paper where the study was actually performed. The injection was in cross-2. So the injections that Peter Strick showed in monkeys going from cross-2 to the STN are the same. In the exact same lobule, we see the hub that changed connectivity during this medication period. So this hub is clearly related to the interaction between the cerebellum and the basal ganglia. Now, the next step of the analysis, since we have some variability within our cohort of 29 participants here, was to see, can we see any link to their clinical scores? So we're looking at similar maps, but what we see at the map here now is not the value of the function of the eigenvector centrality itself, but we're looking at the correlation between the change in the eigenvector centrality and the change in the UPDRS, Uniform Parkinson Disease Rating Scale scores. Those scores, the UPDRS is the classical score for Parkinson's. It involves multiple tests. Part 3 is the part that's being done by a clinician, so a clinician asking the patient to do this and that and ranking them. Part 1 and 2 are questionnaires asking participants about their daily life activity and how the Parkinson's affects them on their motor symptoms and also non-motor symptoms. And while type 3 fluctuates a lot with the severity of the symptom even during the day and from day to day, part 1 and 2, the personal report, are more stable over time. And interestingly, we find better correlation with part 1 and 2 that present something that is more robust over time. And we see it in a few different clusters. Once again, 4 functional hubs. One of them is once again in cross-2 of the cerebellum. Once again, the same area that the work from Peter Strick's lab was suggesting that is directly connected to the subthalamic nucleus. So to summarize this part, we found significant modulation, a modulatory effect of long-term levodopa medication on the basal ganglia cerebellar connectivity, which was very specific to the connectivity between the basal ganglia and the cerebellum. We haven't found a change in connectivity between other nodes in the network. The effect was evident on the network level, so individual connection between individuals, we couldn't find the same level of change. When we're looking at the eigenvector centrality mapping, we found multiple hubs of increased connectivity, and importantly, 2 of them were within cross-2, which we know that connects to the STN. And lastly, when we're looking at the relation between those changes to the symptoms, we see that we can find a positive correlation suggesting changes in the connectivity, changes in the symptom severity related to cross-2. So when the connectivity, when the symptoms get worse, the connectivity increases. So to summarize, and I see that I'm way earlier than time. Sorry, as I was running fast. So to summarize, if we're looking back at the UN heart brain model, model for the brain paper, and their suggestion of the compensatory effect, pathological and dopaminergic effect, I think that we see evidence for all of those in the longitudinal data. And if I try to map it on the timeline of Parkinson, so if we're looking at the normal cerebral function, I would suggest that the compensatory effect comes earlier on, so it peaks before. What we see here is actually already late. The compensatory effect is I would suggest part of the reason that we don't see motor symptoms early on in Parkinson. The compensatory effect help us to delay those motor symptoms, and in that sense actually delay the diagnosis. At the point that we are diagnosing, the compensatory effect is already weaker and is not enough in order to fully compensate and to prevent the visual motor symptoms. The pathological effect then becoming much more severe and lead to the deterioration. And the dopaminergic treatment is slowing it, maybe even reversing it initially, as we see the increase in the connectivity hubs in the cerebellum following the medication. Even 2 years later, we still see this increase. So it may be even reversing it more towards the normal function, but obviously then the pathology kicks in as the symptoms getting more severe in the advanced late stages of Parkinson's. So with that, I would like to thank you for listening. I would like to thank the student who did this work, and I'm looking forward for your questions.
Shlomi, thank you so much. If you have any questions, please go ahead and just raise your hand. I will be happy to call on you. And meanwhile, let me begin by I guess summarizing what I learned and some puzzles that I don't quite understand. So you demonstrated that the patients have higher volume in the cerebellum and thalamus, but then there's this digression, this reduction in volume with progression, and it mostly happening in the white matter, particularly lateral regions of lobule 7, crus 1, and crus 2. And then the second part of the studies were on resting state. And if I understand it correctly, fMRI mostly measures mossy fiber activity, which would be consistent with the idea that STN input to the pons to the mossy fiber inputs are what is largely being affected. And given that on the one hand, you have reduction in volume, particularly white matter. On the other hand, there was this change in the functional connectivity. Do these two things go together somehow?
Well, that's a good question. Yes and no. So once again, what we see that the timelines are not perfectly aligned, and the comparison is not perfectly aligned. So we need to be a bit careful here. But what we see the initial increase, the increase at the baseline, right, which we presumably starts earlier. So that's presumably the compensatory effect that we see that at the time point of diagnosis, as the time point of the initial scan, we see this increase in volume in few regions, including the thalamus, including the white matter of the cerebellum, probably compensating for the reduction in the basal ganglia activity and efficiency due to the neurodegeneration. And then when we're talking about and that decays over time with the pathology. When we're talking what we see in the functional connectivity there, where we see the increase in the, so first of all, when we're looking at the ROIs and the classification, we see a change in the network activity. We cannot say it's more or less there. The eigenvector centrality analysis was added exactly to be able to address it and ask, can we say something about what is this change? And we see specifically this hub in cross 2 that's showing this increase in connectivity. And this increase in connectivity there is presumably related exactly to it, to the accounting for the deteriorations that are happening around it, if that makes sense.
Right, right. One of the other things I noticed was that when you were showing the connectivity results, if you can go back to that that plot that showed there was a clear pattern, in the this it's so interesting. So you said that, I mean, first of all, as you said, in the cerebral cortex, the correlations are much higher, it appears, across regions.
Which is well known. And to begin with, right, there is a question to what extent fMRI in subcortical regions is even interpretable, right? There will be those that argue that we understand MRI, we developed fMRI based on the cortex, right? There we know what we measure. And indeed, yeah, you see much higher connection within the cortex. It's much more correlated. Even after we remove the global signals, everything is relatively more correlated within the cortex. And you can see the separate hemispheres, right? More correlation within each hemisphere. Interestingly, when we look at the cerebellum, you see that you have a high connection between neighboring regions and also across hemispheres, right? So if we're looking at the right 1 to 4 versus the left right to 4, they are connected to each other. So there is some interesting pattern of the connectivity in the cerebellum. Similarly, we see some interesting pattern of the connectivity between the cerebellum and the basal ganglia with some structure within it. So there is some interesting structure there that can definitely be looked deeper into.
There's a question in my lab.
Thank you for the great talk. I was curious about the correlation of the release of the dopamine from the cerebellum on the putamen and also cortex on the putamen. We know that both, given the anatomy you showed us, are connected to the putamen and release dopamine, which is have excitatory and inhibitory on the GPI. I was curious which one is who is leading whom? Is it the contribution of the cerebellum is look like the contribution of the cortex of the releasing of dopamine, or is that different mechanisms? What is your thought?
It's a fascinating one, which I will stop myself from making claim because I have nothing to base them on except from pure guess. So yeah, what leading what? That's a very good question. So I love the classical textbook model of the direct, indirect, hyperdirect. It's so easy to understand, and it's such a beautiful model for Parkinson, right? You can so easily explain what goes wrong there. And once you make it more complicated, right? Once you add the connection to the cerebellum here, and then you try to understand what changes and how does it affect and what lead to what, it just becomes so much more difficult to make sense out of. And I think many more studies, animal model studies, that actually looking at the connections and the effect of deterioration on one on the other are needed in order to really make sense within the circuitry. Because from as far as I'm familiar with the literature, so if we're talking about those models and those findings, we're still lacking the details and the longitudinal information in order to really understand the circuitry and its development. Its deterioration, I should say.
Thanks.
Shlomi, what are your current plans?
Current plans. So that was a side project, to be honest. So that stuff, I started my group during COVID, and we started by working with data sets. Most of the work of my group over the past few years since we got out of COVID was focused on deep brain stimulation and neural oscillations in Parkinson's. So the cerebellum is there as an aim. I want to get to neural recording, neural stimulation at the cerebellum in Parkinson patient. That's something that I would love to do, but we don't have it yet. But in the basal ganglia, we do have deep brain stimulation. So you can turn on, off, you can change stimulation within the subthalamic nucleus and see the effect on the network. So we have a few papers coming out, some of them are already preprint, looking at it, first of all in the clinic. So my push towards the real world, starting by moving from this in the MRI scanner to playing pool in the lab, we push the research into the clinic, piggybacking on clinical session of DBS programming, where the clinician changed the DBS parameters, we record EEG activity and movement with sensors, and we try to map the changes in the brain activity to the changes in the stimulation and to the neuroanatomical location where the electrode actually sits, and to see various insights from the work started by Peter Brown group and other looking at neural oscillation, generalize it to cortical oscillation in a way that is can be measured in a clinical session can be actually translated into clinical practice. And then we also look at the propagation of the beta activity of this oscillation from the brain to the muscles. So that's in collaboration with Dario Farina here at Imperial, that spent decades on developing motor unit decompositions, so we can look at the neural activity of individual motor units and look at the oscillation within those motor units relative to the oscillation coming from the cortex and from the STN, and to see the propagation and how the changes in the DBS affect not only the oscillation within the basal ganglia or the cortex, but also the propagation.
It's wonderful. I'm so grateful for the review that you presented at the beginning of the talk. That was really lovely, as in the context of the research results. If there are no other questions, I want to thank you for the time, and thank you everyone for coming together today.
Thank you.
Thank you for the invite, and thank you everyone.